Okay, good. So very pleased to be here. This is the place I've been visited many times, and I have people like Dick Kirkers and Jack Seller. Those are guys, my mentor essentially. I learned so much from them, and very pleased to come here. And I couldn't made a visit last year because of my health issue. But anyhow, I'm glad to be here. So I will go a little bit quick, and since most of the folks know what I'm doing, the talk I'm giving today, the first part, maybe some students learned from my Tappy Nano meeting, but the second part, I'm sure nobody have heard of, even we published already. So let me, before I forgot the thing, I always typically forgot, the folks involved in this work, two of my postdocs, Xiaoxu Zhuang and Jing Gan, and then I collaborate with Professor Xiao Zhang. He had graduated from UBC. Dr. Sadler is his advisor. And my colleague is at the Forest Products Lab. So our lab is pretty small, and about down to about 150 people, maybe 50 scientists, and it's fully funded by U.S. government. And it's been there for a long, long time. And we've been early days in the Wisconsin campus. There was my later point. Okay, here this building, 1910, and then we moved to this new building in 1930. This building composite lab was established in 2010. So. I'm trying to go over a little bit of some other work I've done before, and many folks probably know this project we did with the biojet conversion work. We did it probably 10 years ago almost, yeah. So we essentially work with the USDA funded project with a bunch of folks in the northern Pacific Northwest, led by Washington State University. Jack actually is on the advisory board of the project, and we were able eventually to convert 6 tons of forest residual into jet fuel, and Alaska Airlines actually flew a flight from Seattle to Washington, D.C. So it's a sort of interesting project, learned so much of it, and had a lot of fun on this one. And then after that one, sort of biorefinery related, and we continue on working on biorefinery. One of the processes we developed is called acid hydrotrophic fractionation. The work's published in 2017 Science Advances, and later on we found another acid, malic acid, even performs much more interesting. We published in Green Chemistry 2020. All these work has not been highly visible because it's the pandemic. So, but I've been presented a couple of times at some other conferences, but folks have interest can look into these papers, and some of the things are kind of interesting. So the ligand we produce from malic acid is really nice because you convert to aromatics. Actually, Professor Shannon Stoll at Wisconsin, he compared our ligand, which is this one, with the GVL, the black circle here, looking at conversion if it's pretty close by, okay, in terms of aromatic yield. And I did a Sanders on crystal work, and I think most students are familiar with this work a little bit in a way. I presented many years ago at the ACS meeting. Essentially what we did essentially is, the CNC production is started within Canada. Professor Gray and the earlier pioneer work in this area. And one of the things they found out essentially that the sulfuric acid concentration, 64%, temperature of 45 degrees C, 45 minutes, produce the best CNC in terms of morphology-wise. So that's what you typically see from literature publication, that they are looking like this. About 100 nanometer long, 10 nanometer in diameter, okay? But the only drawback is the yield is very low. Typically get a 35, 50% at most. We were able to identify, if you do it at a lower acid concentration, not so much low, just about 4-5% lower. So down to about 58 to 62%, you can get a much higher yield. Oh, sorry. Much higher yield. Also, slightly different morphology. For example, in this condition, S stands for sulfuric acid concentration, temperature, and time. You get a 70% yield. This is based upon a total fiber. Okay, this is bleach pulp, so cellulose content 85%. If you based upon cellulose base, this yields more than 65.9%. And if you do this condition, we obtain 72%, which means about 90% cellulose yield. So that's what you get. And moreover, the morphologies are very different than what you saw before. So these CNC can have advantages for certain applications. Particularly if you try to make a film, we'll give you stronger films. By doing so, you can also tailor not only the yield, but also the sulfur content. You see, we sort of map out what the sulfur content will be by doing different hydrolysis conditions, okay? And then we start from there. We say, oh, okay, if you don't do over hydrolyze, most of the folks do it because don't want to deal with the issue of fibers, then you actually can improve yield. And think along this line, you can use a little bit of weak acid. So in this case, we do dicarboxylic acid, much weaker as compared to sulfuric acid. You also can produce both CNC and the CF. So the yield is very low, but most of the material, if you go to fibrillation, produce the fibrils, which is just as nice application for things, different areas. And if you don't separate the CNC from the residual fibers, you just fibrillate, you get a sort of more like a CNC-like materials, you know. So, and some of you probably may know I work with Liang Bing for quite a bit in the last 5 years. And he's the guy leading a group. He published a paper in Nature called "Develop Super Strong Wood." Essentially, you densify the wood to, for example, the height was 44 millimeter down to about 10 millimeter, so 5-fold reduction in terms of volume, and you get a very strong wood. You know, for example, here the natural wood is a green bar, a blue bar. This wood about the red bar in terms of tensile strength-wise, so like a 10-time fold increase. And later on, we also published a paper together in Science, looking issue that how to make a wood as affordable as a paper. So this is exactly published in 2021. My contribution is really to work with him to develop a delignification technique, because he started with, he's not in the water area, he started with the alkaline sulfite, which we know is kind of older process and have a lot of issues in terms of environmental impact. So we work with him to develop a little bit more nice chemistry. In this case, we use traditional oxygen delignification, which was used in the paper industry for pulping, right? And typically used for removal of residual lignin, but we apply to remove the bulk lignin in wood. Now, it has two advantages. First of all, that the bulk lignin is much easier to remove because it's not condensed. The residual in pulp is much harder to remove. Second is that typically in the industry doing 14% into auto-delig. The reason because pump. You can't pump thicker than 14%. It's a little bit more difficult. So the issue is that oxygen solubility in water is low, so the efficiency is low. But doing bulk delignification, you can run reaction in gas phase, the efficiency is much higher. So in this case, we just run the traditional pulping conditions. It works beautifully, and we're able to produce similar results as Liang Bing did initially with alkaline sulfide delignifications. For example, I think this is our wood after delignification and pressed. This is his wood after delignification and pressed. Look at the tensile strength, almost identical improvement. So that's pretty much all the other work I have done so far. And today, this started with today's talk. So it's actually looking into the cells of microfibrils, or called microfibrillated cellulose production. I focus on two areas. One is looking at the high solid production, try to understand the mechanism. And because right now, all these commercial instruments, machines does 2%, so a lot of water involved, which is a huge problem in terms of transportation and in terms of water treatment, okay? So our objective is later, first of all, looking at the solids loading effect on CMF production. Second, compare the treatment, for example, either using enzyme or mechanical fibrillation, refining, if that improves fibrillation. This project is funded by US P3 Nano, which is a private-public partnership. Then we look in this produce MFC for barrier coating. In this case, just initial study using banana. The characterization-wise, we're mainly using light microscopy and SEM because there are different ways to characterize MCF, but it's very, very difficult to get a very good information of it. We did some study on water retention value measurements and the sedimentation suspension stability, looking into the properties in terms of these materials. So, okay, so here is the schematic. Essentially, the pulp. In this case, we're using commercial birchwood pulp and just feed it into mechanical refining through treatment, then do ball milling at high solids, up to 28% solids, or you do enzymatic treatment and go to ball milling, and or you don't do any treatment to compare, see if the treatment has helps or not. And the reason we choose ball milling because it's very hard to find a very nice device can run at a high solids at a small scale for laboratory studies. So we just say, okay, that's for our first study, just look at the ball mill, see if that, what happened to it. And then we're using these optical techniques to look in the morphology of the materials. So this slide captured the essence of the result. So we did a solid loading 1%, 5%, 70%, 28%. Okay, this is at a time 0.5 hours, so 30 minutes into bone marrow. And you see here that at the low solid content, you see the fibrillation here very clearly. Okay, and same thing you see at a 28% some fibrillation, but you don't see anything at a 5% and 17%. And that was really a surprise to us, and we didn't believe it. So we go back to look more of these samples. It could be the sample issue. So we look at so many pictures, we found this is exactly what happened. At 30 minutes, you don't see nothing fibrillation at a 5% loading, 70% loading, but you do see fibrillation 1%, 28%. And if you go 1 hour, you see the fibrillation continues for all the samples, and particularly 70% now you see fibrillation. You see fibrils here. And 5% some tiny ones, okay? And then you go to like 3 hour, 2 hours, 3 hours, you see the fibrillation much more, and also you see a lot of fibrillation in the 5% and 70%. Actually, the internal extent of fibrillation, the 70% does quite well at the high extent, longer times. And so these are the contrast higher optical images to see what they look like. So obviously now you see 28% actually the fibrils is thicker than the 17%. You know, the dark color really means so fine you can't see anything. But here you can tell the bigger fibrils. So this is a little bit of puzzling. Initially we had a problem trying to understand what happened to it. So, and then we did so this slide summarizes what really happened. It really means at the earliest stage, nothing happening into fibrillation at the lower or medium solid loading, but there are some fibrillation at the very low or very high solid loading. And as time goes by, this high solid loading effect seems decreased. Essentially becomes this monotonic trend in terms of extent fibrillation with the solid content. So we did a water retention value measurements because water retention value measures the amount of water absorbed by the sample, means how porous they are, or in a way maybe more of micro and nanofibrils in there. So same thing, shows the same results that early stage fibrillation, these two samples as a low and a medium solid loading has a lower water retention value, but at 1% and 70% very high. And as time goes by, this effect goes away, become monotonic curve. Essentially high solid loading has low retention value and the lower has the highest, okay? And we check with the suspension stability, shows the same results. So here, the first of the set of samples at the early stage, 0.5 hour, and the last set of samples at 3 hours, later stage. You see here that the early stage, the curve goes like this, right? That means these samples much coarser, so sedimentation goes much faster, and even at 8 hours, shows the similar trend. But the later stage fibrillated samples, immediately you see this sort of monotonic trend goes down, decreases the stability-wise. So these results, both the water retention value measurements and the sediment test, agree with the optical observations. So we explain this as two mechanisms. One is the primary mechanism, which is just the balls and the fiber impacting interactions. This is the primary mechanism causes the fibrillation, but there is a secondary mechanism, interfiber fibrillar tensional or frictional interactions, and that happens only at high solids. So de Kerkers did this long time ago. He found this called crowding factor, right? So essentially, in order to form fiber flock, you need certain concentrations. And based upon his work, we can calculate what is the minimum concentration needed to get a fiber flock. So using his number, suggested number, crowding factor 60 to 130, we calculate the minimum concentration is 1.4% to 3%. So obviously 1% is below this number. There's nothing flocculation happening there. There's no secondary mechanism at all. But why even 5% don't see the effect of a fibrillation of secondary mechanism? We believe that this is just the minimum requirement to form flock. But in order to have enough force created by the tension or friction force, the flock should be much stronger. So the N has to be much larger than that. So that's why until 17%, we see sort of secondary mechanism comes into play. So then after this work, we did a treatment, look at the enzymatic treatment, and I think Jack did bunch of work in this area too. We essentially found ionometry is quite effective. This is the exact same thing we found out. Mechanofibrillation also helps in a way to fibrillate the samples. And these are just the results. You see that. One of the things we found out is here. This sample is untreated. After 2 hours, the extent of fibrillation is similar to this sample treated with mechanofibrillation, only 0.5 hours. So the initial pre-treatment by mechanical refining is very effective for nanocellular production. So in summary, basically, we have found two mechanisms in fibrillation. One is just simply the ball and the fiber interaction is the primary one. Second is the fiber fibril tension or frictional force that causes secondary mechanism. And this mechanism becomes less important as go to finer refining. So it doesn't help much in nanofibrillation, but does help in microfibrillation. So with that, we moved to this project, try to coat in banana. I learned this from essentially the MBAN folks. They did the carrot nanocellular coating the banana. So we said, okay, if they can do carrot, what can we do fiber? What are fibers? So that's where we started this work. We're not going into detail. So in terms of coating method-wise, you can do spray coating or do dipping. For laboratory study, we just do dipping study, which is much easier to do this. So this is a very simple schematic. Essentially that you have bleach pulp, mechanofibrillating, and then you make a certain concentration, and then you dip the banana into it, the whole thing, including this end. This end is very important because this end that the ethylene gas released from the banana to ripen the banana. So important to make sure to coat the whole thing. If you go to stores, you'll find out they wrap it with plastic films to reduce the amount of release of ethylene gas. And then we're also making a casting film to look into mechanical properties as well as the gas permeation property of these films. This just shows you what these materials look like. The one pass through the supermoist colloidal versus the full pass. Obviously, this material is much finer, and this is much coarser. The first step we did is to look at what's the concentration used in coating. To do so, we did essentially a contact angle measurements. So if the contact angle less than 90 degrees C, that means the banana can be wetted. So that means the coating will stay. If it's more than 90 degrees, probably not desirable. The coating will go away. It won't stick to the banana. So eventually we found a 0.75% concentration is good concentration to coat it with the banana. So this is sort of the image we see. So okay, the first is the control, day 0, 2, 4, 6, 8, 10. And pretty much by day 4, they're starting to have browning color, which is not desirable for saline anymore. Right? And you coat it with the microfibrillated cellulose, seems to reduce the browning a little bit. For example, here compared to here, right? Either you compare, I mean, the finer the fibrils, the permeation is low, so obviously the reduction is also more. This improvement is not so significant, but it does improve a little bit. These are the coating film on the banana surface, what they look like. We just take a small piece of it, look under the microscope. So obviously the fibrils fibrillate more, you have more finer things, so it's less porous. And so then we look at the weight loss. Weight loss is very important because essentially the grocery stores sell by weight. So if you lost 10%, you lost 10% profit right away. And that typically happens all the time. Any fruits they bought to the store after a few days, the weight just reduced. So they lost money right away. So they wish to sell as early as possible. So looking at the control, which is the star, about 14% reduction after day 4. Day 4 is the day that we say the banana start to browning, so you don't want more than 4 days. But if you coat it with the microfibrillated cellulose, depending upon the fineness of the sample, the reduction can be significant. For here is under 8%, so you reduce by less than 6% weight. That's a pure profit margin for the grocery stores. Okay. The second, we look at the firmness, because once the banana becomes so soft, then usually means they are kind of too much ripened and it doesn't taste good. So the softness reduction also is significant dependent upon the extent of the fibrillation of the microfibrillated cellulose. So for example, let's see this one. This is day 4 again. The control is the star right here, and this is the full-pass flow microfibrillation is right here. So which is measured by the force we measured by doing a punch study. So this is about 6.5 Newton. This is about 8.5 Newton. So you see the banana keeps its firmness. And then you look in the soluble sugar in there, because that's a taste measure too. And if it's too much sugar, that means the banana is fully ripened. You don't want to fully ripen too early, right? When it's still firm. So here, the control is about less than 20 whatever this number is, total soluble solids is 20%. This is about 70%. So the coating has effectiveness in terms of to reduce the soluble sugar, keep the firmness, and reduce the browning of the banana. And then we look at the film, what it look like, because the customer still want to see what the actual banana look like. If it looks kind of goofy on the surface, then they don't want to buy the thing, right? So the film, these film are pretty transparent. So, I mean, obviously you guys all know this, right? And this is just then we're looking at if this film is washable. Because one of the things with apple is coated with something. I don't know what it is, but it's hard to wash out. We know that, right? So this one, we look at how they're easily washable. So what we did is just measure measure the contact angle of the film with the water. So you see all these contact angles less than 90 degrees C, then they're wettable by water, which means they can washable by water. And the film, we also measure some the cast-emitted field. You look at their water vapor permeability. So for example, this is the water vapor transmission rate, and you can see here, this is a different kind of microfibrillated cellulose. The 4 means the fine ones, and the 1 means the coarser ones. So obviously, the fine ones, the density is high, and the water permeation also low. But we also can look at, for example, how much you coat on it, right? So you can coat it less, like as low as 6 grammage-wise, 6 grams per meter square versus 20, which is pretty thick film this here. I mean, certainly increased grammage, it does prevent the water permeation. It goes down. But, you know, if you look at the cost-wise, if this is an issue that 6% versus 20%, 3 times more of microfibrillated cellulose, so costs much more, 3 times more in terms of raw material-wise, but the gain is not that much. So you rather use a finer microfibrillated cellulose than using more microfibrillated cellulose in this case. So as I mentioned, the first study we did is purely microfibrillated cellulose, nothing else. That's it. So here we added some additive in here to see will they help in terms of prevent water permeation, in terms of prevent oxygen permeation and ethylene permeation. And mainly we try to look into if it improves browning, because browning is the measure that customers use for purchasing decisions, right? So the previous one we said some improvement, not a whole lot. Here now you see is a pretty good indication when we added some additive. This is control. This is the pure MFC, and these are different additives we added in there. You can see here that these are 2 full days after the study. These are 6 days. They look at the color are very different. So we delay by exactly 2 days in terms of browning white. But then here, here is the, let me see, these are 0 days, and this is the 4 days. This is 4 days. So 2 days delay of browning. And we look at the 7 days, obviously very different. So we found that these additives are very effective. So we also apply this pure microfibrillated cellulose and this new formulation, whatever, into this fruit you probably never see before. It's called star apple. We're working with the University of Florida. They have a fruit farm. And this star apple, obviously, is a small quantity production in Florida, but it's quite popular in South America. So this is no coating. You know, after 4 days, it's already kind of shrinking the skin and dry out. And this is coated with pure microfibrillated cellulose. The shrink is not as much as this, and this is coated a little bit different formula. We look in this outside skins, you can see the performance in terms of different days are very different. And then we cut out, let me see what I see. Oh, this thing didn't come out, unfortunately, this figure. We cut out to see the fluids itself, actually. There's a quite a significant different improvement in terms of with the coated materials. With that, I go very quick, 25 minutes, okay? So leave plenty of time for you guys to ask questions, yeah.
Thank you, Jay. Before moving to the next talk, maybe we have a chance to discuss the presentation, the results. This is very practical, very relevant as well. So we have a question there.
Yeah, did you test the efficacy of the film compared to the plastic wrapping around the stem of the banana?
Yeah, we haven't done that yet. That's a good question. We should actually test that one first of all. We also should test next time is the cold temperature storage. But most of the fluids, when shipping in the shipping process, they always put in the cold storage. We can test that one because the issue with the cold storage is just energy cost. So we should do that too in the next step. Yeah. So our first year project just looking if there are different fluids. Next year we'll do a little bit more careful study. The issue is fluids you see and know, so you can pick one time. That's it. So yeah.
One other comment to this is the fact that from banana peel you can produce fibrillated cellulose.
Yes.
That would be super nice circularity.
Right.
Banana peel
Right.
to produce the MFC that you need. Yeah.
Great talk to your wife. Is this different technology than Capitec? Sean Ireland's.
Yeah, we don't know.
MFC.
They must have a patent. I don't know. I didn't ask Sean last time I saw him in terms of what exactly patent he patents. I know John Simonson from Oregon State patented nanocellulose-based coating. Essentially, it's a CNC and maybe CNF. So my angle initially thought, oh, the nanomaterial is nice, but it's too expensive. So that's just local microfibrillated. And I didn't realize Sean Ireland actually had a company working on banana until later when I did the work. I realized. So I don't know what patent they have. And we right now file a provisional with Washington State, Xiaozhan doing that thing. And what we're trying to, from different angle, maybe we're definitely going to add an additive in it, not pure MFC, because the performance is different, yeah.
I assume it's John Simonson's work because
okay. So that's,
on the Capitec website it says
okay.
State in 2020.
So in that case, just the nano material.
It's fibrillane MFC.
Yeah,
Oh, okay.
It's definitely MFC though.
And then
Yeah.
with John
Yeah.
Simonson's-
That's with the clay as well, right?
Not anymore.
No.
Okay.
So we'll see that in terms of the patent office, what they say, those kind of things. But we essentially try to work a little more other than just pure MFC, maybe add something, because particularly antioxidant and antimicrobial properties can be useful for the packaging of things.
But I'm surprised that it's actually a good barrier because it's porous. So oxygen gets out or ethylene or whatever it is. So how does it?
Yeah, so the issue is this. We did this, so now we can understand a little more than a year ago when we started the project. So turns out you need the respiration. The fluids, a lot of things in there is live. You need the respiration. So for example, banana, if you coat it too tight, what happens is produce ethanol because it become an anaerobic digesting process. Produce ethanol, that tastes bad. So you need some respiration. You need some reduced permeation, but not completely eliminate. If you eliminate, you produce ethanol. Yeah.
Sure, but then the gases are getting in and out. So why isn't it why is it changing the degradation rate? It is. I just don't know why.
Yeah. So you want a slowed aerobic digestion process. You don't want an anaerobic digestion process. That's what you want. You want it slowed, but still aerobic, not anaerobic. Yes.
Emily, and then we go here. Yes, Emily.
Thanks again, JY. But so along those lines, I gather that you don't want to tell us the secrets of your additive, but what's the role of the additive? Is it like an antioxidant, or is it changing barrier properties, or what do you want to use it
So
for?
you want, I mean, you can add a bunch of things in there. You want it as simple as possible, also safe, because you talk about food, right? So particularly in the future, we're probably not going to do banana, the reason because U.S. don't produce much banana. So that's the main reason. So you want something simple and has multi-role. So ideally you want antioxidant and maybe some antimicrobial properties and also reduce gas permeation. Because I try to use MFC, not NFC. If for NFC, you probably don't care the permeation anymore. It's pretty good already. So.
That was a very nice presentation, and so it's exciting also to see applications of cellulose. I was curious in terms of actually how much material do you think you get deposited per banana?
It's very small. Remember the gravity, we talking about 6 grams per meter squared.
Okay.
So it's very small, yeah. Yes.
Is the intention to apply this prior or after the ripening process?
So this will be applied right after harvest.
Okay.
Yeah.
Yeah.
Okay.
Over.
Have you looked at what that looks like? Because the banana is bright green when it's harvested, right? So like.
Yeah, so that's a problem. I mean, I showed some pictures. Yeah. So the issue is that these are pretty green when we get it from store, but even after 1 day, it's already becomes kind of yellowish color. Yeah.
So what is the time between harvesting and when it's up in the store shelf?
See, I don't know. I mean, I would think like 2-3 weeks. That's why with this project we're gonna work. These bananas from our local supermarket got it, but we're gonna work with University of Florida. Actually, the fruits they pick day before and they ship it to us. So these fruits actually got it from Florida. They just pick it and the next day ship it to us overnight. So it's only maybe 2 days the most. Yeah.
We have a question there first.
Yeah.
You used bananas as the main produce crop for this study, and then we saw your work with apples. Is there a criteria for picking crops you think work particularly well with, things that we typically peel and don't eat the peel, or is it something you could say like other bananas, oranges, melons,
Yeah.
or things where we get the peel like apples?
Yeah, excellent question. So the issue is here. First of all, right now, USDA only certify banana can coat it with cellulose materials because they can peel it away. So we have to work with the FDA and the USDA to look into other fruits applications. We know apple already coated with something, so we're trying to work on apple too. Now, second is fruits considered 2 types. One called climateric, another is non-climateric. So non-climateric, probably the coating doesn't help much because it's already, when you pick it's completely mature. But climateric, you always pick a green because there's a process, ripening process, right? So most likely gonna apply to the climateric fruits. So.
Hi, Yuan. Amazing presentation. Just curious, if you're trying to commercialize this technology, so generally there is not just one banana. So how do you think that this technology can be commercialized, or do you have idea about it? Like, you can't dip one banana because in reality there are a bunch of bananas and they are not even separated, So do you have thought about that process? Like,
Yeah,
would be
it's a good question because the banana has a whole hand, right?
Exactly.
So there's something, yeah, that's a good question. But I think dipping would work in this case if that's the case because there's plenty of space between bananas. For dipping would work, but spray wouldn't be a problem. You're right. Yeah. So different fruits may have different method. Yeah.
There was a question there.
Yeah, we talked to just comment on the FDA regulation of the cellulose. Can you comment why FDA is not approving the cellulose? I know the nanocellulose because the nano still on the separate chart, but for microfibrillated cellulose, really is microfiber. Can you comment on that?
So I think there is not just, they basically need data. That's all they need. So we need to have to do a study to make sure this is safe. The reason banana was okay is that, Junyong's company is, what's the Junyong company name called? Very Advisor. They work with FDA on that banana certification process. So essentially they need the data. That's all. Yeah.
Maybe one more question, Adam?
Very exciting work. Just a question regarding the volume in terms of, since you're doing a lot of fruit packaging at a larger scale, so right now you're using microfluidizer for the final packaging. I'm just curious, doing at larger scale, do we need some other equipment to produce MFC that can be suitable for these?
Yeah, so this is produced from the Japanese, whatever that device is, right? So University of Maine has a sort of 1 ton a day capacity for that process. I'm sure MFC produced by other companies like Sappi and Beauregard should all work. There's no reason why it should not work. But Maine is going to develop a facility to produce photo-grade MFC. So that will be useful, those applications. Yeah.
Maybe one more last question.
Yeah.
Very last one. Okay, 2 then.
Do you think differential dipping time will have any effect?
Can you repeat the question?
Do you think differential dipping time will have any effect on it?
Different?
Dipping time.
Oh, dipping time. Oh, it's very short. Yeah, it's very short because the suspension is pretty fluid, so it's very wet, very quickly.
So how much time do you
I think only a few seconds, then we just move up. Yeah, yeah.
Jack, very final question.
Very nice topic.
Yeah, drying is an issue. So right now we just hang it in the room, and we don't care how long it dries. But that is the issue you have to consider, because for packaging house they have to, don't have long space for them to just dry, let it dry. So that's an issue you have to consider. Yeah.
Super. Thank you. What do you think, JY? This is a very smart crowd.
Oh yeah,
right?
I know. That's why I speak fast. They understand this.
Yes, yes. Thank you, JY. A combo of presentations. It was a short one, but very meaningful. Now we have the second presentation. I haven't introduced our speaker, Philippe Amstislavski. Sorry for my pronunciation, but he comes from University of Alaska. It's a short flight from Alaska to Vancouver, from Anchorage to Vancouver, which is very nice. He is a professor there of public health. Very interesting individual. We were walking yesterday. We went to the beach. We went also to the Museum of Anthropology. It was wonderful, and I realized that you are pretty knowledgeable in the indigeneity matters. So I encourage you at the end of the talk to ask questions about indigenous efforts in Alaska. He's very closely connected with that, and I think we can learn a lot from you in that area. But today he will be talking about mycelium. Maybe there is a connection as well, but the floor is yours. Welcome.
Thank you. Thank you, Orlando, for inviting JY and myself here. We appreciate it. I live and work on the traditional lands of the Dena'ina, Tlingit, and Haida people in South Central Alaska, so just up the coast from you. We share the same biome in both regions. We are also at the edge of the temperate rainforest there. The tree species, the animals, and even the fungi are similar or the same. And I think we are sharing some of the same challenges in our coastal communities. And to illustrate that, I wanted to share some of the background information on plastics and plastic debris in Alaska. It is of concern to our Alaska Native communities because in some of our villages, over 80% of protein comes from the land and from the sea. So subsistence is not a matter of recreation. It's a really matter of livelihoods for many families. So plastic pollution it's a direct concern to Alaska Native communities and fishing communities along the coast of Alaska. Our students last year did a transect on the coast of Gasku Island, which is also known as Forester Island. That's actually very close to the coast of British Columbia. And it is surprising what we found. We found that about almost half of the pieces of plastic by volume come from construction industry and from fishing industry. So there's bigger chunks of plastic. They come from either the wall insulation or from buoys and fish gear that got lost or got kicked off board during fishing, commercial fishing operations. And then the rest of the stuff has to do with buoys. They're harder plastics typically, and hard pieces of drift nets that have been lost or discarded, and hard plastics and rope. In terms of the impact so materials for construction and materials for fishing end up in our water stream, and eventually they make it to the Gulf of Alaska and to the Pacific Ocean. Now, in terms of the carbon footprint, EPS, which is the almost half of the debris by volume in the transects we did, are coming from EPS specifically from this one type of plastic, expanded polystyrene, is only second in terms of carbon footprint to steel in all construction materials. So after steel, that's the second highest contributor to to carbon emissions in the world coming from industry that has to do with packaging, cold chain shipping, and construction. We then said, well, I worked in northern Alaska with the Alaska Native communities for several years, and as a public health official, traveled to communities that are off the road network. We have about 600 villages that are located off the road network. So you can only fly there, or you can take a boat. And so the transportation is expensive, and bringing things to rural communities in general to Arctic communities is very expensive. So there is a saying that what comes to Alaska stays in Alaska. And it has kind of ironic implication. It means that all the stuff that we don't know what to do with ends up in the landfill. And our water and garbage management systems, waste management systems are very, very simple. It typically looks like a pit in the ground. And it's then covered with soil once it's filled. And so the pits unprocessed garbage, so everything goes there, from human waste to styrofoam containers, from fishing to your wall insulation once the houses are broken and discarded. This is actually very telling image. So we're just landing at the village of Shishmarov, which you may have heard of this community in the news. It's one of the communities that has to relocate within the next several years up about 7 kilometers up into the tundra from this island, because it's getting inundated because of the sea level rise. So there is very small difference. And you can see it's very, very shallow, but it's also very flat. Because of that this whole entire community has to move upland, and so we are in the direct path of climate change effects in the Arctic, because 1 degree temperature difference is the difference between melting ice and snow and stable ice and snow. So that is something that I always mention to people in temperate regions, So this is the transect from Gasco Island. You can see the different things that we typically find with students. So the EPS chunks, that looks like wall insulation at some point, or was wall insulation. This one looks, it could be a side of a cooler. It's hard to tell because they're all beaten up by the current. And a lot of those other pieces actually have to do with buoys. We have to have buoys to keep the nets afloat, and those buoys are often lost because they break off when if you get weather on the boats. When you pull them out, the buoys often break. The connectors from the buoy that hold the buoy to the net break, and then you have this floating thing that's there for a long time, and that's. From a different site, from Kachemak Bay in Alaska. These are pieces of extruded polystyrene floating in, actually there is a seal colony there. Now, in terms of the back to the wall insulation applications, in Alaska, and actually across the North American continent, the housing stock is about 40 years old on average. That's the median age of residential house in North America. In Alaska, it's a little older. I think it's a little younger in British Columbia. The housing is younger. Most of our housing has been built in the 70s during the oil boom. So all of those houses were built with not a lot of thought about energy consumption and embedded energy. And they are in bad need of retrofits now, energy retrofits. And it is a real issue for us. We pay very, very high price in rural Alaska for energy because the diesel seems to be the primary source of heat in many communities, diesel and firewood. And it's very expensive. Many households spend half of their income in Alaska Native communities, especially half of their income literally on keeping the house warm during winter. So retrofits are really, really important economic matter. And when we peel these walls, you get to see what's inside. You get a lot of rot on the inside of the structure. The rot is mostly mold that's grown because the houses were built improperly. So the water vapor, when it moved in, we have a lot of temperature differential. Of course, it could be minus 40 on this side and plus 20 on the other side of the wall in the house. So when you have cold air touching the surface of the warm wall, then of course you get condensate, and it starts creating these perfect conditions, like a perfect petri dish condition for microorganisms to grow. So you get a lot of this stuff. And when we retrofit, this is how it typically looks. So it's a secondary wall that is EPS with 2x4 siding. And so we frame it, and we attach it to the outside of the building. And then there is usually metal or vinyl siding that goes on the other side. And so you have double walls in this retrofits. And so we said, well, can we challenge this? Can we come up with a material that can perform on par with synthetics, but can have less or no carbon footprint and can be produced locally? All of our styrofoam foam, all the insulation, and all the fish shipping boxes are brought to Alaska from somewhere else. Washington State produces a lot of styrofoam, and it comes on these massive barges and we mostly ship in air, right? And so it's very expensive by the time it gets to Alaska, and over 90% of the cost is actually the shipping. So what we said is, we created, when I started at the University of Alaska, we created a small collection of cultures from alga, from several species of alga to different polypores, basidiomycetic fungi that grow in circumpolar north. And then I was lucky to go to Finland and work with VTT Finland and start developing methods to how to introduce cellulose and cellulose feedstocks into those composites. So if you remember your biology 101, so the mycelium is kind of the root network of fungus. So it's similar to what the plants do with their roots. Right, so it's out there. It's vegetative form of the organism. It's looking for nutrients, and converting organic matter into food for the fruiting body. Now, this structure is three-dimensional. It permeates pretty much every inch of the square. Every micron of rainforest floor. So that property is really interesting. When we started adding cellulose to it, what we ended up with is very dense slab of material that had some interesting properties, but it was too dense, and it was not insulating enough. But had some interesting properties with respect to hydrophobicity. It was not easily wettable, because we knew because we knew that these organisms produce hydrophobins. It's a type of protein that repels water, and also had some antimicrobial properties, which you also knew, kind of expected to see, because, well, penicillin is a fungal product, right? So those properties were kind of expected, but not really defined or characterized in this type of materials. So this was the first composites that we produced on the previous slide. Then we said, well, what if we start foaming cellulose and create this really lightweight matrix of cellulose fibers intertwined, and then we add mycelium or culture to the blend and actually let it sit and incubate for a bit and see whether the mycelium will have an easier time growing through this kind of matrix, because it's less work for the organism to split those fibers to get to the sugars. And so we started foaming, and when we add the biosurfactant, what happens is that you have this big things, the cellulose fibers, the cellulose bundles, and the bubbles, as they form, they attract the species of mycelium that has been added. So they form on the inside of the bubble, and the fibers start intertwining into layers as we foam. So when you push the air into the column, it looks literally like a massive vat of, say, cappuccino, right? It's the same consistency. We then apply vacuum to densify it, to control density, and we let the material incubate for a bit. And that bit depends on how thick the slab is, but we've done anything from, so this samples over here, they are about 5-day incubation, and some, and here they're 10-day incubation. So it's, actually can pass them around if folks would like to see them, maybe like this. So that density now decreased, and then we said, well, can we use locally sourced locally sourced source of cellulose, or cellulose that's from our own trees. And in Alaska, there are about 1.6 million acres of beetle-killed forest now, especially in southeastern Alaska. And that front of beetle kill is actually advancing every year northward. So now it's around, I don't know how many people have been to Alaska. Not many. So one. You guys are so close. That's surprising. Yes. Well, you're welcome to come and visit our lab. We will also show you the beetle-killed trees, because they are right outside of my lab. And this is mostly spruce. We have a lot of spruce and hemlock. And they're 0.6 million acres just out, around Anchorage. And then another million acres of beetle-killed forest in central Alaska along the coast. So a lot of those trees, they are standing right by the road. They are fire hazard, and they fall often. And so we have to remove them. When they are cut, they basically just left to rot on the side. So we started processing them and it's a very simple process. We debark. And once we debark, we make chips, and those chips go into a disc refiner, and we produce MDF. With we wanted to produce least amount of side stream from this process. So MDF seems to be the kind of the good compromise here. We wanted the lignin to come out of the cell walls and flow. So that MDF actually allows us to do that, because it's heated enough so that the lignin is now released. And then we add mycelium culture during foaming process, and that slab goes into incubation. And what I'm passing around is actually the product of about from 2 to 10 days incubation, depending on which sample you're holding. So that's as far as the process. We then started experimenting with different kinds of structures, from hexacomb structure, just cardboard fed to the fungus, to spraying on the surface, add it into the blends. And so what we found is that the best results in terms of thermal conductivity, density, and hydrophobicity we actually get when we grow the material we add the inoculant into the blend, into the cellulose pulp and surfactant blend, and let it incubate. And we do it with spray. What we get is we get a mass of fungus growing on the surface. And so because it doesn't know which way the nutrients are, right? So it doesn't have ice, right? So it's chemically sensing the environment, but it takes it a while to decide, well, to sense that there is food source here. So eventually it grows through and grows downward, but it takes it a while. So in terms of the kind of economy of time, this seems to be the most effective process. So that's a schematic of the whole process flow here. And this is how this looks in terms of the production. So it's a small scale production at the moment. Nick here, whom JY trained in Forest Products Lab, is doing the mixing. So he's actually adding in inoculant right now. And then that's the slab that's ready to go into incubation. And what I passed around is what it looks like when it comes out of the incubation. And this is some of the applications under the ARPA study that GY and colleagues at NREL and I are doing. So this is the interior of the exterior of the building that I described. This is that added retrofit on the facade. So mycelium cellulose foam goes in here, and that's the old insulation that has been here from 1970s or whenever the building was built. And these are some other examples of how we envision this can be applied. Packaging, when you talked about bananas when they come to Alaska, they look already like day 5 or 8 in your slides, because they are shipped from Seattle usually. Wherever they come from to Seattle, they sit there for some time, and then they go on the barge and into ferry, and then by the time they get to Skagway or to Sitka, it's a couple of weeks later, and so it was very relevant to us to increase the shelf life. The packaging is a big issue, but we also use over 1 million boxes of styrofoam boxes in Alaska. We are the biggest producer of food or seafood in North America. So most of the biggest export from Alaska is salmon and halibut and ground fish. A lot of it is going to out of Alaska in styrofoam boxes. So we started working with a couple of indigenous communities on the coast, one on Kodiak Island and one on Kenai Peninsula, looking into replacing styrofoam. They're very concerned about the styrofoam being present in the coastline. We know already that there are animal health implications of those of the microplastics being ingested. We don't know yet what the human health implications are, but we suspect that once we do know, that it will be quite late in the game to start coming up with solutions. So thinking forward, when we created the slabs, one interesting thing that mycelium cellulose foams provide is that they're still biologically active unless you dehydrate them or do something to the organism that renders it dead. Essentially, you kill it. It can be fused biologically. So instead of using adhesive, you can press the two panels together and they fuse. So the mycelium recognizes itself, and the hyphae basically start intertwining and interconnecting, so you get these nodes, and it's quite strong. And so we have tried it by just pressing boards together, and so you can get different densities on the top and the bottom and the middle. So you can have a very strong finish. And I passed a piece of mycelium leather in that box too. Yeah. So that.
I thought it was a tape.
No, it is. Nope. No, it is. It is 100% mycelium product that has been coated and dyed. So depending on how dense it is, how much pressure you apply when you're growing it, and then how you post-process it, you can get some very, very different results. So it can be flexible or it can be very stiff, depending on how much you press it. So those are some of the kind of examples of how we envision this could be used in the future. In terms of the properties, so we have tested 5 different organisms with the same cellulose blend and hexacomb. So the thermal conductivity in this first paper, the one that we just got accepted, was actually somewhere just below 0.4. So now we were able to improve it quite a bit. We're up to styrofoam thermal conductivity. So we are around 0.5 on average in our samples. So that's the work that JY and I have been doing. The thermal conductivity and density, of course, are interrelated, right? So there is dynamic relationship in the two, but the denser the material is, the higher thermal conductivity would be. So that's something you would expect with most materials, especially with open cell foams. And then in terms of the compression testing, they don't differ tremendously in terms of compression testing, except for the hexacomb material. That one was very poor, and we think it's because the fungus actually it kind of delaminated it and removed the glue that was used to make the hexacomb material itself, because it's 2 layers of paper essentially of kraft, and then the hexacomb structure, a mesh in between. And then water contact analysis was, so it actually, the surface turns out to be very, very, very low wettability. So the water contact angle was from 120 to 160 in some samples. And then we noted also that it doesn't really drop over time. You would expect it to decrease, but it doesn't seem to change that much. And so the Tremes versicolor is the turkey tail. That's the common mushroom that you have here as well. Fomes fermentarius you don't have in this region. You do have it in British Columbia upland. It's the birch polyp, or it grows on birch. There's quite a bit of work actually by VTT on that species specifically. Irpex lactus you do not have here. And Schizophyllum commune you should have. Ganoderm lucidum also is present in some parts of British Columbia. And Pleurotus astratus is the oyster mushroom you can actually find in the stores, I think. And on this side, these are the species. These are the actual treatments. So control foam with, and then you can see that just foam itself has the cellulose foam itself, we know it doesn't have very good water repellency properties, right? So it immediately sags and gets wet very quickly. And then when we add mycelium, it goes up to 140. Degrees water contact angle. And this is something I wanted to share also. This is when we add, so this is the mycelium cellulose composite. dropped into water, and we're spinning it. So you can see it's starting to absorb water, but it's buoyant. And think of buoys, right? And things that we use now that are made out of styrofoam. And it doesn't break up. And when we go to just cellulose foam, so cellulose foam itself is very hydrophilic, and it swells immediately, and then it sinks and it starts breaking up very quickly. So that's kind of that's an important functional difference. We also looked at, I mentioned the antimicrobial properties. So we tested 6 different species of organisms that are pathogenic to humans, 5 species that are pathogenic to humans, and MS virus, which is corona group virus that's kind of used as a model for, it's a virus that is not pathogenic to humans, but similar to corona group viruses. And what we found is that in all of those organisms, there is a selective ability to deactivate the virus and has some. So that the blue one is the MS2 virus, which is the one we were most interested in. And we also see that, so if it goes that way, it means that's a log increase over 1 million. So anything that's over 1 million, that's statistically significant. Anything that's going that way, it means that it does not affect the microorganism growth. So things that are on this side are interesting to us. Staphylococcus aureus is typical, it's a human infection of the lungs and skin that's quite common. And it's a typically hospital-transmitted infection. And so that is one species that does, Fermentaris pinicola has a very distinctive ability to suppress this Staphylococcus aureus growth. And so that was really interesting finding, that it's selective. So it's not suppressing all the pathogens, it's suppressing some. And the gene expression and the mechanism behind it is not well understood. We don't actually know why it's doing it and how. Is it some kind of surface phenomenon, or is it metabolites that the organism is producing, or the combination? I don't want to bore you with the process flows, but this is just the overall diagram of how this works in terms of how the production process works. And this is the structural properties. So this is cellulose you're well familiar with. I don't think I can teach anybody anything useful here in this group about the cellulose properties. But if you look at this sample here, so this is the same cellulose. This is control, cellulose without mycelium. And this is mycelium that's feeding on that cellulose. So this is the same, so the same slide, but there is only one tiny piece of cellulose left after 10 days of incubation. So the fungus basically bioconverted all the cellulose into a biomass. So it utilized almost 100% of it, except this little piece here. And that is a shot on the surface. And when you look at the surface, the reason it's white like this is because it creates this mass of hyphae that's aerial that's going up. And it doesn't have any cellulose in it. It's just a layer of pure mycelium on top. So the inside of the material is quite different than the outside in terms of the morphology and structure. So yeah, these are some main findings, just the water repellency that I mentioned, the insulative properties, the physical strength, and then selective microbiality. And I think the most important one actually in some ways for our communities is the fact that it biodegrades in the Arctic environment, in the subarctic environment quite well. So this is after 6 months of the samples being buried in the soil. You can see the roots, spruce roots going through it, and leaves being embedded there. Squirrels actually eat it. We don't know exactly why they like it, but I think it's about 25% protein content by dry mass. So I think they figured out that it's a good snack. That is something that was critical also to our work. And we all standing on shoulders of thousands of years of Arctic tradition of innovation and in biomaterials. If you think of kayak, if you think of the sunglasses we all wear, right? These are Arctic innovations, right? And they were all produced in the Arctic with, they're very functional. We use them until today. So there is a lot to learn, both from natural environment and from the indigenous communities that we are surrounded by. And these are some next steps. Prototype 1 is what we've just finished. We are now looking at additives to make it less fire prone, or adding fire retardant materials to it. Because it's cellulose, of course the cellulose will eventually burn. Chitin chars very well; it doesn't burn but when that initial layer of chitin is gone, then of course you have exposed cellulose fibers that will ignite. So we are looking into adding different additives. That's one area of inquiry. I think that folks here have a lot insight into, and we do not. I'm a mycologist, so this is not my forte at all, and I would love to collaborate on this work. We're also looking at we are producing 200 square feet by I think our milestone is October. So to produce that amount, the reason we are making so much is because we want to apply to a building and actually test it in Arctic under Arctic winter conditions. We are looking into e-spinning and creating filaments. Expression of genes that are responsible for hydrophobicity and antibacterial properties is something that would be very interesting to study. And development of mechanistic models that explain actually what we know now to characterize these materials, but we don't know the mechanisms behind some of the processes that we are seeing. So exploring those mechanisms would be, I think, a really good way to go. And thank you, everyone. I also thank you for our collaborator. JY traveled farther, actually, than I, times 3, I think, right? It only takes 3 hours to get here from Anchorage. And our collaborators at VTT Finland, at NREL, National Energy, Renewable Energy Laboratory. And thank you, Orlando, for inviting us here.
Thank you, Philippe. I love this science. It's simple, it's impactful. I think we need to see more of this, right? The science is behind all of this, but in a way, creating this type of impact is really, really very relevant. By the way, to the community here, the BPI, one of the first investments we made in BPI was to acquire a foam-forming unit, the same that you use in VTT. You saw that yesterday. We have that. And we have also radio frequency drying. Drying is very important. That would be one question that I wanted to ask, how you dry your foams. But anyhow, we have those capabilities here, and we have also faculty here like Johan and other teams working with mycelium. So this is very relevant to us, and I think this is an opportunity for us to also interact with you, because we're close and we can share, in a way, quite synergistic interactions. One very brief question. You have there a lot of protein, so can you eat that foam? Is it animal feed or can be eaten?
So this particular fungus is, yes, it's edible. we've done bioassays. It's not toxic to humans. It doesn't taste good.
Put the orange flavor on and put it in here.
Well,
Here we are, yes.
The taste is like tempeh. It's not great. So nobody likes to eat. Well, very few people like to eat. Actually, my kids like to eat fresh tofu, right? But I don't think it's a common thing, right? So it tastes. It doesn't have a appealing taste. It's not sweet, but yes, it is edible. We actually brought some chips for JY to try, and it is something that can be eaten. But I think that it's high quality protein,
yes.
So I would think that human consumption is probably higher priority than animal feed in this case, and it would be interesting to explore. Yeah.
Yes. So besides proteins, you have a lot of chitin in this material, right? So this is quite interesting. So let's go for questions. We have about 10 minutes for questions. So this is great that we can sort of have a conversation with the speakers.
Thank you so much for this wonderful talk. Because I saw that you mentioned that squirrels were eating this kind of material, you said that it's edible. So if you're using in construction, will the rats make their colonies inside those, I don't know, facade or something like that? Is there a problem of rats?
Well, so fortunately we don't have rats in Alaska. So that's one good thing, but there are rats in other places. I don't know about rats, but I do know that the squirrels are attracted to it. So, we talked about it quite a bit because, yes, in principle, a squirrel should not be able to get into your wall. If it can get in, it's already a problem. But they are very clever animals, and they can chew pretty much through anything. So especially our red squirrels, like the climate change heroes. They march north every year. They love climate change, and it's really interesting. We have red squirrels now in Fairbanks. When I moved to Alaska 11 years ago, there were no red squirrels. So they are very adaptable, and I think that adding something that tastes awful to them would be the way to go, like menthol or something that really, really repels them. It's a good question, yeah. Thank you.
There's a next question over here.
Yeah. So I have a question. Great talk. Yeah. So I have a question regarding to the board that after finishing, do you need to sterilize them? Because in principle, the fungi are still growing, the mushroom is still growing, so with time it consumes the cellulose, right? Degrade the cellulose, the foaming like structure.
Yes.
And also they can generate spores on the surface, so potentially there can be some kind of concern. Safety?
Yeah, that's a very, very good point. Thank you. So the species we use is it takes very long time for it to actually produce a fruiting body that can sporulate. So we remove the board from incubation. The longest we keep it in incubation is 10 days. So 10 days is not enough for a mushroom to produce a fruiting body. We do not sterilize because it's a very energy-intensive process. It's very expensive also, and what we do is we just dehydrate the board, and that's, I think Orlando was going to ask that same question. How do we? So that's so we put it in a conventional dryer with exhaust, and we just dehydrated it at 50 degrees centigrade for 24 hours. So it just dehydrates the board, and when it does finish, it's about 3% moisture content, and so it's very low moisture. And we've tested it. The mushroom does not recover from that kind of moisture loss because the cell walls just collapse. There's just not enough water in those boards. So that's what we have been doing, yes. But if you don't sterilize or dry, it will continue growing, and so eventually you'll have a block of chitin in your wall. So that's probably not great.
We have one question here, one there, and then last one, maybe John.
So obviously great innovation, and I know this is probably further down in the future for the project, but I'm just wondering what your production costs were like for this material and how competitive it would be to market compared to styrofoam, which is relatively cheap to produce.
Thank you. This is the time, that you say you don't know, right? We don't know. That's the truth. And that's because the costs are very low now because you add very small amount of cells to the blend, and we use salvaged wood. Now, we don't have pulping facility in Alaska. So we have to send our spruce to JY in Wisconsin, and then he pulps it and sends it back to Alaska. So not a very sustainable process, and that's because we don't have a process to pulp, and there is no one pulp mill in Alaska. So that is, in principle, the cost will be the cost of energy to process the wood, so to chip it and pulp it, and then of course human labor to harvest the wood and debark it, and then the incubation process itself is hands off. It just it's a biological process, so you don't need to do anything. It's as long as you set the correct conditions. And it's the handling and cutting the boards into smaller pieces. So so I know I'm not answering your question, and it's not it's not because I want to duck it. It's just because we don't have these answers. That's something we are working on now with LCA and with NREL, trying to actually understand the costs of production in Alaska and how to produce the feedstock in Alaska that can actually be turned into, fed into this material process. Yeah.
Now, maybe I know, foam forming is industrially viable and it's being produced in companies in Finland, in the US, and in Sweden. So as long as you can produce layers that are not as thick, you can make multi-layers in principle and have something that is economically more viable.
Yes.
The continuous processing in fast speed fabrication, that is already here. So
yes, it's existing.
There is quite promise here. So Lorenzo.
Yeah. Thanks for the great presentation. I have a question regarding the processing and also characterization. The first question is that what is the mechanical strength or compressive strength of these foams that you're making and how far it could be improved? And the second one is that how we can change the porosities inside these foams, for example, governing them based on specific levels, or what kind of processing parameters you're changing to achieve that. And the final one is that have you tried to make aerogels out of them, means ambient pressure dryable aerogels, or ambient pressure dryable aerogels out of them?
So thank you. These are really good questions. We don't have the facilities to work with gels. As Orlando mentioned, the technology foam forming technology is mature. It exists already in the world. It's very effective. So we know how to produce foams. We also know how to change the density. So that's to answer your question about the density, as we can apply more or less vacuum into the chamber, and we have a knockout chamber. So the liquid it's extracted, it's going into the chamber, and you can then recycle it because it has still the surfactant, so you can use it several times, up to 3 times actually you can use it. So that is how you can control the density, and you increase or decrease it. You also can control the thickness of the slab, so regardless of the density, by cutting it or by just pouring less or more material into the mold. There is a paper that should come next week in Cellulose, and I can maybe share a link to it or give Orlando the link once once it's published, so that you can look at this, because there's several types of composites. So I would just suggest looking at the different composites and their compression properties, because they differ. So it varies from the compression strength of this material is close to styrofoam foam, to expanded polystyrene, but there are some that have higher compression because the way we've produced them. So yeah, I would look there, just so in the interest of time.
So we are already over time, or close to finish. Two quick questions here, right? And then Philippe and JY will be available in the afternoon, so they will be around.
Yes. We're here until tomorrow, so
yes.
we want to talk to us. It's easy to find us, yeah.
Hi.
So 2 questions here.
Yeah, thank you for your presentation. I'm always interested in the seemingly contradictory trade-off between degradability and longevity. And right now, compared to your control, the thermal conductivity is not that great, but what is improved is your water contact angle and your antimicrobial properties, and yet you can claim that it's biodegradable. So like how does that work? Like what are the mechanisms there?
So I would defend the thermal conductivity because it is on par with styrofoam. So it actually is quite good now in this last set of experiments. So we've matched styrofoam 1-to-1. And that was the kind of the benchmark, because that's the synthetic incumbent. I think it's a really good question about how you balance biodegradability with longevity. So biodegradability is, it's an amorphous concept, right? Because it depends on where and how do you biodegrade something. So marine environment has a completely different biota, completely different salinity and pH than freshwater, a stream, or a soil. So I think that we need to qualify it more carefully, and that's something that I think as researchers we have the responsibility to do it. Where biodegradable where? So our tests are in the taiga, in boreal forests, different biota than here, different microorganisms. So that I think is really key to think about what is the environment you expect this material to end up with in. Is it going to be a rainforest, temperate rainforest? Is it going to be sub-Saharan Africa? Is it going to be coastal marine environment in Alaska? So I think that's really important. So we have not studied that. We know only about soil degradability at the moment, and that's something that we need to do going forward if we want to really understand how these materials behave. And I think that's something to do. I invite you to be in touch, and maybe we can come up with a way to set up an experiment under this ARPA award, because it would be really good to test it in coastal Alaska, coastal British Columbia, and inland, rainforest environment versus taiga, to see how that changes.
For single-use materials, this is ideal, right? So you have the example of salmon packaging, right? Yeah. This is really very nice application in that sense, right? If the biodegradation is not really there. So last question here.
Great talk. More of a comment or maybe a collaboration opportunity. Rand's point about them still being active. Well, can't we utilize that to make a self-healing material? So that if it gets wet, you put it in a wall, if it gets a little bit wet, it starts growing again, it seals that back up. The hyphae give a hydrophobic outside coating to it, and it's better. So you could have a smart healable foam.
I should show you this experiment we did last week. So when you break the board, when it's still living, and you push it back together, it regrows within 24 hours and seals itself. And it's actually the seal where the crack was, it's quite strong. So I think you cannot probably use it in that way in the wall, right? Because you have to have some moisture in the matrix. But you could patch cracks that exist, and then basically remove the moisture from the wall just by dehydrating it. So it's as opposed to say using polymeric foam that's used now. If we could use that, because we pump our material, and it comes out the same consistency as the foam you would get from this gun that people, in construction industry use a lot to seal holes in building, cavities. So I think that's something that would be really important to explore as well. Thank you.
Perfect.
Yeah,
Thank you.
Definitely a collaboration opportunity. I'll take you up on it.
Super. Let's give an applause to the speaker. We should take a picture. If anyone wants here, let's take a picture with the speakers. But before you leave, tomorrow there is another talk by a researcher from ETH. He's coming to talk about protein microfibrils. So continuing in the living matter subject. So that will be tomorrow at noon. Subs also will be served. So you all are welcome. I really appreciate your attendance.